Hello students, today we are going to discuss sexual reproduction in flowering plants part 2. So in part 1 we earlier discussed about what are all the organs reproductive organs in the plants. Then we just discussed about gametogenesis. So gametogenesis is production of gamete cells. Gamete cells is the reproductive cells which are important for reproduction, sexual reproduction. Then after the gametogenesis, the cells are produced, the reproductive cells are produced. Then the reproductive cells have to meet each other. So for this, pollination is an unavoidable one in sexual reproduction. So in pollinations we discussed about catenogamy, xenogamy and the autogamies. So through these pathways the pollinations will happen in plants. So today we are going to discuss after the pollinations. So after the pollinations, the plant involved in fertilization. After the fertilization, the zygote will produced, then embryogenesis. After the embryogenesis, the seeds will be formed, then some different kinds of seeds and some more technologies in seeds we are going to discuss today. Okay, so let's get into the topic. So here guys, so after the last part we just discussed how the gametes were produced in male and female part of the plants. So the male part is the androecium and the female part is the gynoecium. In androecium, in the anthers position, the pollen grains are just produced. So here you can see two different kinds of pollen grains in generally seen in plants. In about 60% of plants have the first kind of pollen grains which only have one generative cell in it. So only one generative cell in it. So it will be only two nucleus cells inside the pollen grains, and remaining 40% of the plants have pollen grain with two generative cells and one vegetative cells formations. So these kind of pollen grains are produced in the plants. Then in the other case, in female reproductive organs, so inside the gynoecium, inside the ovary, and specifically inside the ovule, the embryo sac is formed. A 7-cell 8 nucleus stage of embryo sac. Here you can see that this one is egg cell, the important one for fertilization. Then near that you can see the synergid cells, and over the top you can see the antipodal cells, and in the center of the embryo sac you can see the polar nucleus. This is another set of cells involved in fertilizations. So this is how the plant is prepared for their fertilizations. So after pollinations, the pollen grains just reaches over the stigma of a plant. So pollen grain is a male reproductive cell, stigma is a female reproductive part, one of the female reproductive part. The stigma is just over the top of the stales. So after that stigma, the nucleus cells, the generative cells have to reach the egg cells. So here we're just going to discuss about the pollen stigma interactions. Okay, so here you can see some figures are there. So you can see that this is a gynoecium part. So here in the top you can see some amount of pollen grains are just tapped over the stigma. Here some pollen grains are grown inside the style also. So in reality, here you can see some microscopic structures. Here you can see over the stigma more amount of pollen grains are dusted over there, and some pollen grains are just ground. You can see some dark yellow patches over there, a fiber like projections from the pollen grains. Fine, the pinky structures are the pollen grains over there. So how the pollen and stigma interactions are there? This is the most important part of reproduction. So these interactions are species specific. These interactions are species specific. That means you can't expect always the same kind of plants pollen grains will be stored in the stigma, or it will be dusted in the stigma. It will be lots of unnecessary pollen grains also just dusted over the stigma, because you can't expect the insects to deliver like a delivery boy an exact address. Okay, so the pollinating insects or any kind of agents, water or a wind or an ants, they just deliver all the pollen grains to everywhere is possible. Okay, so you can expect different kinds of pollen grains, different plants pollen grains are just dusted over the stigma. Here the pollen grains, the surface of the pollen grains and the enzymes present inside the stigma are species specific in nature. If they were from a same species, means they will accept each other, so the enzyme will cleave the proteins and it will let the growth of the vegetative cell. So here we call it as a tube cells. So it's nothing but the cell present inside the pollen grain. This is a vegetative cell. So here we call it as a tube cell because it grown like a tube-like projections from the pollen grain to the embryo sac. So these enzyme and that surface proteins are just a species specific elements. They not only reject the other species, sometimes they also reject the same plants pollen grains. So if the organism is self incompatibility, so if an organism is in self incompatibility, means these kind of organisms rejects their own pollen grains if it is from the same plant. So that means they were always preferring the cross pollinations. Fine. So if it is a self pollinating pollen grain, means they will reject those things also, and also they were easily rejecting the different kinds of plants also, plants pollen grains. So that's why only a same kind of pollen grains were accepted in a stigma, so that there will be no collapse between the genetic materials naturally. Fine. So this is how the interactions begins. Then after that, so just consider the pollen grain is accepted by the stigma since it is in a same species. Now so if this is a pollen grains, means you can expect two generative cells and one tube cells. The tube cell just begins to grow through the pore in the pollen grains. So it just started growing inside the stale. So you can see that this is a stale part of the stale part of the ovary, stale part of the pistil. Right, stale part of the pistil. So inside the stale, the tube cell just going through it. Okay, so how it grows? So what feeds it to grow? Means it's just growing by absorbing the sugar materials present inside the stale. So there will be some sugar materials are there. These are the chemical elements which guided the tube cell to grow until embryo sac. So the tube cell recognize the chemical composition of the sugar substance and they absorbing it for their nutritional element, and they just growing inside the stale. So these kind of growth are called chemotrophism. So sensitized by a chemical element and growing towards the chemical element for an irreversible growth, that's called chemotrophism. So those kind of movement just happening inside the stale after the pollen and stigma interactions. Fine. So this is how the tube cell grown inside the styles, and it just growing until reaches the embryo sac. Is that clear, guys? Fine. So the next thing, how it reaches the egg cells. So as we already discussed, the tube cell is growing, then after that the genital cell will move into the tube cells, and they just moving continuously until the tube cells reaches the embryo sac. So there will be another thing is there. So as we already discussed before, some plants there will be two generative cells, in some plants there will be only one generative cells. So what happened to those kind of plants? So if it is a only one generative cell means, what happened to those kind of cells means, so after the tube cells are growing, the one generative cell enter into the tube cell, and during the movement time, the one generative cells mitotically divided into two cells. They just mitotically divided into two cells. Okay, so during the movement through the tube cells, it just growing, it just divided. Okay, fine. So after the tube cell, so here in this figure you can see the tube cell is just continuously growing until reaches the embryo sac. Fine. So after it reaches the embryo sac, the generative cells, that means the male gamete cells, also just following the path and just try to enter into the embryo sac. So here you can see another microscopic images where how the tube cells growing inside the styles. So clear, guys? Okay. Now, so here is a structure of the embryo sac. After the tube cell grows, it just reaches the embryo sac through the micropyle end, of the ovule. So this is how it moving. So inside the vegetative cell, the two generative cells also follow the path. Okay, so this is how it just meets the embryo sac. So after it meets the embryo sac, there will be three possibilities of movements. In most of the cases, the tube cell just grown inside any of the synergid cell, either in the right side or in the left side. So either of the synergid cells, they just penetrated inside the generative cells, so that it can release the generative cell inside the synergids. Through this synergid path, the generative cell will reach ultimately to the egg cell. So this is a one type of movement through the synergid cells. Then another types are, it may move either between the synergid cells and the walls, between the synergid cells and the walls. This is another one type. And the third one type is between the synergid cells. So these are the three variations. The tube cell just grown inside the embryo sac and deliver the generative cells. Okay, in different plants the different kinds of approaches will be followed. Fine. Now the fertilization process. So here in these images we can see that the male gametes are just delivered inside the synergid cells, so that the male gametes of generative cells can reach the egg cells. So you can see that egg cell is just near the synergid and the polar nucleus is in the center of the embryo sac. So there will be two fertilization process are there. So we will call it as a double fertilization. So first thing, there are two generative cells. So let's write generative 1 and generative 2. So there are two generative cells are there. The first generative cells move inside the tube cells. They're just going to reach the egg cells of the embryo sac, which is just above the synergic cells, and they just fuse together. So in this fertilization process, they just formed a zygote. So it's already discussed that the gamete cells, the male gamete and the female gamete, they are just haploid cells. Haploids means one set of chromosomes only present inside the cells. So after it fused and get fertilized to form a zygote cells, so just it has a combo of male and female cells. The genetic materials is doubled again a 2n normal diploid cell. So which is going to develop into a embryo in future. So the next process is the generative cells 2. The another generative cell is just going to reach the polar nucleus cells. So polar nucleus cells just in the center of the embryo sac. So after it get fertilized with the polar nucleus, so the generative cell is haploid 1, but the polar nucleuses are a single celled and double nucleus cells. So there will be two cells inside the polar nucleus, so there will be two 1n cells are there. So here the generative cell is a 1n and the polar nucleuses are two individual 1n cells. After it get fertilized, after it get fused, so it formed a 3n cell. It get a 3n cell. So this fertilization we call it as a triple fusion. Triple fusion. Since we just have three individual haploid cells fused together, we just calling it as a triple fusion. So this triple fusion will result in the formation of endosperm in the seeds. Fine. Endosperms are just food materials for growing embryos. So we will discuss in our later topics. So this is how the double fertilization process occurred. Fine. Okay, let's move on to the next topic. So here is embryogenesis in plants. So you can see the fertilized egg in the initial process. Fertilized egg is just a 2n cell. Initially get fertilized the first generative cell and the egg of a embryo sac. So after this fertilization, the zygote, a 2n single cell, they just mitotically start dividing into two cell stage. Then once again it will get a multiple stage, that means a 8 cell stage. So here you can see that a part of cell is just settled over the down and most of the cells in the upper region. So you can see that head-like projections and the flattened neck-like projections also there. After that, it form one of the important stages, a globular stage. Here you just start forming the embryos and cotyledon type of cells. After that, the triangular type of cells. So based on their shapes, they just giving the names. After that, a heart shape of cells. After that, the torpedo shapes of cells. Here it will be almost like a complete embryo. Then a matured embryo with enough amount of cotyledon materials and embryonic materials. So this is how the embryo is getting matured during the seed formations. After the flowers get fertilized, the accessory parts of flowers just removed and the ovary part is start growing into a seed and fruit molecules. Seed and fruits. Fine. So this is how the embryos are formed inside the seeds. So as we already mentioned, the fertilized egg just grown into an embryo and the triple fusion, the results of triple fusion, the 3n cell, just grown into endosperm. Fine. So endosperm is like an for a nourishment for growing embryo and seeds. So even after the seed germinations, the baby like embryo part just need additional food nourishments. They just get it from the endosperm parts. Okay. Fine. So now the embryos are generated. So here you can see how the embryos will form in two different kinds of plants. The first one is a dicot and the second one is a monocot. So here you can see how the embryos part developed. So from the embryo part we can see the plumule region as marked in the figure. The plumule is a part which is just going to grow into a shoot, so over the soil. So after that we can see the cotyledon regions. So the cotyledon regions, you are just seeing that there are a broken cartilaginous membranes like things. So you can see the entire embryo is there. Here two parts were like a small partitions you can see. So you can see that two number of cotyledons are there, but still it is in same embryo. It's not like a two individual. It's just broken off at some part. Okay. So that's we call it as dicotyledons. Fine. Then under the cotyledon is a hypocotyli. Hypocotyli is simple, but simply think as lower the cotyledon regions, which favors for seed germinations. Then after the hypocotyl you can see the radical part. Radical part is just going to grow as a root. So after the radical part you can see a root cap over the radical region. So you can see that the root cap is an important part which prevent the baby radical from mechanical damages or any kind of injuries while growing from the seeds. Then over the top of the plumule, there will be a protecting layer for plumule also, that we call it as coleoptile. So the important aspect of coleoptile is it recognizes the light, it just sensitized by the light and just make the plant grow towards the light. So it favors the phototrophism. So this is how the embryos grown inside the seeds. Here you can see a monocot seed. So here coleoptile, shoot apex, that is the plumule in dicot regions, epiblast, radical part, root regions, root cap, then coleorhiza. So coleorhiza is the initial part of the shoot root regions. After the coleorhiza only, so after crossing the coleorhiza only, the radical grown into a root. Fine, just a protecting layer. Okay, so this is how the embryo just grown inside the seeds. So here the next part, some types of seeds, how the embryos present inside the seeds. Here you can see cotyledonous structures, where the cotyledonous structures present inside the seeds, and some examples like monocot seeds and the dicot seeds, how it formed, and where the endosperm will presented. So as already mentioned, the endosperms are nourishment for them growing embryos and growing seeds. So outside the seeds, the fruit will be formed. The fruit is just nothing but the ovule part, the outside the ovule part. The entire ovary is just grown as a fruit regions. It was generally the plant grown, but here for these fruits they are just simply different from other organisms, they just form a false fruit, a pseudo fruit, because so you can see here is the seeds, so only the layer outside the seeds are the fruit, so these regions are the fruit regions, these regions are just the fruit regions, so they are only from the ovary part, ovule part, outside the ovule part. So the remaining things are just the thalamus like things which are not really fruit materials, but these plants adapted like that. So here also this is the real fruit part, but the edible part is not really a fruit regions. So we generally think that this is a fruit part, so we call it as a pseudo fruit, a false fruit regions. Fine. Okay, so these are some examples of seed formations. After the seed formed, it just dispersed by either hydro or wind or any kind of animals. So after it just dispersed, it just start growing through the radical part at first and the plumule at second. Fine. Some different things in the plants. The first thing is parthenocarp. So parthenocarp is nothing but a seedless fruit formation, a seedless fruit formation in the flowers. So this bypasses the fertilization process, so that the fruit productions will be automatically started inside the flowers, and but the seed will not be produced since it sees the process of fertilizations. So how it occurring means, in naturally it will occur in different types. So first one is based on the environment, specifically in lower temperature than the optimal one. So if it is a lower temperature means, due to lower temperatures, that fertilization process were not much active, but after the during the time the fruits process will begins after the lower temperatures. So say for an example, so for a period of time due to lower temperatures, the fertilization is just ceased for a time, but the seed formation did not wait for the fertilization process, then it automatically formed. So since the fruit productions are started now, the fertilization process is completely and seized one, so the fruit produced with either minute seeds or completely out of seeds. So this is how the fruit productions formed the parthenocarpic process. So not only in environment, it can also triggered through plant hormones. So we call it as growth hormones. So growth hormones also triggering the parthenocarpy process. Fine. So this is one of the economically important step. So most of the people prefer a parthenocarpic fruits, so that there will be no disturbances with the seeds. So these are economically important techniques in agriculture. Then the next thing is apomixis. So apomixis is completely different from parthenocarpic, so don't confuse with this. So apomixis is the formation of seeds without fertilization. They just formed seeds formed without fertilization. So how these things occurred means, so you all know that initially inside the ovule, the megaspore mother cell in the female reproductive organ is generally in a 2n cell structure. After the meiosis cell divisions, it just formed 4 1n cells, and 3 of them are degenerated, and one formed embryo sac. So this is how the female reproductive cell produced in the plants. But in this case of apomixis, it just directly bypassed the entire process and just formed a fertilized seed productions. So that means directly enter into the embryogenesis. So the two N cells directly enter into the embryogenesis. Here it has to be entered into the meiosis process and formation of embryo sac and fertilization by the pollen nucleus cells, that is generative cells. You can directly write it generative cells. So only after the generative cells, just wait. So after the bondage with generative cells, after that only this gives a embryos through the embryogenesis process. So now in apomixis, it bypasses the entire steps and directly enter into the embryogenesis directly from the 2N cells in the ovule. So by this way it just produces seed. It is simply a asexual kind of reproductions and ultimately the fruit. So this is how the fruit formed in apomixis. So here you can see that the entire process of meiosis and pollen transfer and embryo and fertilization, everything is not needed here. So there will be, this is really economically efficient for a plant to produce. So plant needs lots of water for these kind of process, meiosis, that embryo sac formations, then fertilizations. Here in apomixis, even in a low amount of water, the plant can produce seeds. So this is also economically important one. Then the next thing is, in apomixis, the plants also produces a polyembryonic seeds. So polyembryonic seeds is nothing but the 2N cells directly involved in seed productions. So it produces multi seeds in a ovule. So multi amount of seeds in a ovule, so that it just produces a fruit with lots of seeds. So you can see some flowers that these kind of fruits producing lots of seeds in it, but it's simply a single fruit. So these kind of fruits formed by polyembryony. The polyembryonic seeds formed based on the apomixis. So these are all about the seeds formations. So hope you guys understand the concepts. If you have any queries, means you can ask it in the chat messages. Thank you.